Thanks, Debashish, and the other comrades in arms as here. And until a few days ago, I didn't realize that I had to give 3 talks. So I hope you'll indulge in my rather garbled version of these 3 talks. But it's also nice to actually come out in the open after being constrained at home or for the last 2 years and seeing people in 3 dimensions. So thanks again for that, for the opportunity to do that. So apart from giving these, my lectures are largely going to be about compartmentalization. And, but what I want to, I also of course, I'm talking to the converted already. But oftentimes when one gives talks in such public forum, especially in India, one needs to convince people that studying the phenomena of life, like cell biology and biological systems in general, is not only a valid exercise, but is actually an absolutely beautiful branch of physics and biology. And so, you know what? One of the features that I'd like to put across in these series of talks is that the cell is just not a random collection of molecules, but it's a very highly organized collection of molecules, very precisely organized within its volume. And it's remarkable that this cellular matter, which is highly organized and only works if it is in this organized state, is also has another very interesting property, namely cells beget other cells. Okay, and this is, I mean, it might seem, this biblical statement might seem not very profound, but please do think about it. You cannot construct a cell de novo. Okay, a cell comes from another cell. A living cell comes from another living cell. And this is an absolutely deep principle in biology, part of the so-called cell theory. And I'd like to invite you to think about the following question, a question that has been posed by many people. Most recently, I heard from Thomas Lequi, who's a colleague of mine, and but Shashi Tutupalli and I have been discussing this for a long time. Namely, the following: if I take a cell, like a eukaryotic cell, and just mush it up, okay, but you do a careful mushing, so you don't break the polymers and so on and so forth. Yeah, but you generally mix up the system and leave it as it is. Will you get back a living cell? Will this cell be alive? And so I'd like you to think about that. I don't know what the answers are. What do you think will happen? Any thoughts? You can randomly say whatever you want. Huh? What? It'll be alive. Okay, most likely. So, okay, anyone to counter that? It will not be alive. Then that's the only other possibility. So, yeah. Now I'm going to break up the organelles also. I'm going to do everything. Yeah. The polymers, I won't break covalent bonds and so on. So I'll also mush that up. So I'll break up all low energy organizations in the cell. But I won't break up the proteins and I won't break up the polymers and so on, because that's held by some stronger backbone. Okay. So anyway, there seem to be many different things. My own feeling is that it won't be alive, and most of the people that I talk to suggest that you might get some membranal structures because of self-organization, but most likely they won't be bilayer structures. They might be multilamellar structures. So what I was trying to get at this is that even if this collection has all the molecules of life, yeah, they still won't be living in my and many other people's opinion, because they don't have the correct organization, whatever that means. Yeah, I don't know what organization means. I don't know how to quantify it. I don't know how to measure it. But somehow there is this magical organization in the cell that imbues those set of molecules to be living. Okay? So that's a profound thing. And now just imagine what does inheritance mean? Inheritance means, of course, molecular inheritance, right? Genetic inheritance. But is that enough? Do we need to, do we need to somehow inherit the organization too? Right? Is there some minimal organization that needs to be transferred from the mother cell to the daughter cells? In fact, what does organization mean and how is it to be measured? You know, that's I think, a deep problem in biology, which I'd like you to think about, both in terms of theory and in terms of experiments. So, yeah. So this is what I'll be talking about, trying to build on the mechanics of self-organization. But the mechanics of self-organization in the cell and in living systems is fundamentally different from the self-organization seen in the inanimate physical world. Okay? And that's if you don't get anything else from these set of lectures, I'd like you to take back these questions and address them. Okay. So what I'll do in the 3 lectures. I mean, the nice thing is about giving 3 lectures is that you don't need to try to finish everything in time. So these lectures might spill over from one to the other, and there'll be some fluid-like object here. But I'm hoping to cover the following. In the lecture 1, I'll be talking about the phenomenology of compartmentalization. The kind of phenomenology that appeals to a physicist, okay? Because I'm a physicist, as many of the people here. By the way, before I get into this, how many of you have training in biology, and how many have, let's say, in biology? Okay, and the rest are physics, is it? Okay. So most of the people are physics people. Okay. Okay. So I'll be talking about the phenomenology of compartmentalization, and the second half of the lecture will be on kinetics of non-equilibrium self-assembly. Okay? Lecture 2 will be on first the mechanics and then the hydrodynamics of the same thing, of non-equilibrium self-assembly, with particular emphasis to Golgi, okay, which is a compartment within the cell. It's a beautiful compartment. It's one of my favorite compartments, and everyone has their favorite cell and favorite compartment, and that's my favorite compartment. The last lecture is going to be on function, namely information processing, and how that influences or dictates or drives the number of Golgi cisterna or Golgi compartments. Yeah? I'm going to loosely use the word cisterna and compartments to be the same. Okay, a sort of stricter biologist would not like that, but let's just, for pedagogical reasons, let's just call it as a compartment. Okay, so the last lecture will involve some little bit of information theory, and I hope to be able to reach that point, because I'm quite excited by that part of it. Okay. Oh, I don't need to go there. Okay. Yeah. So the lecture 1 now, I'll be talking about compartmentalization in eukaryotic cells, and I'll try to show how the logic of compartmentalization sort of invokes the idea of cell as a distributed computing system. Okay. Then I'll be talking about the non-equilibrium physics that is relevant for compartment formation. I'll be talking about time, space, and energy scales. And I'll also talk about the active mesoscale process of transport, fission, and fusion, and how they are driven by activity. Then I'll be talking about non-equilibrium's assembly of compartments, which carries a finite current, finite flux of cargo or vesicles that flux through the system. I'll be initially talking about the control of structure, spatial organization, and chemistry of the Golgi cisterna, or Golgi compartments. And finally, I'll be talking about non-equilibrium assembly of compartments, again with the current, but here I'll be talking about how to achieve size control. Okay. There might have been lectures of size control in polymeric systems. No? like in cilia or something? Oh, the near is given. Okay, so then it will be complementary to that. Yeah. Okay. So before we step to this thing, there's a famous quote of Dobzhansky, which says, "Nothing in biology makes sense unless viewed by the lens of evolution." So this is the required slide that is to be put. So this is the tree of life, or the bush of life, or whatever you would like to describe it as. And oops, yeah. And what you have here are the archaeal branch and the bacterial branch. Okay, and these, of course, this is this time scale here is, let's say, 4 billion years. And the eukaryotes, with all their compartmentalization, which includes our cells such as that forms our body, they form through a marriage between the archaeal cells and the bacterial cells. Okay, in the process called eukaryogenesis. And it's this remarkable organized matter. It's not to say that archaea and bacteria are not organized. They are organized, but to a lesser extent than what we have in eukaryotes. They come about because of a marriage between the archaea and the bacteria. Okay? So there's an emergent, compartmentalization is an emergent feature of eukaryotes. And so this is here the modern view of a very dynamical cell. Yeah? This is, of course, a static picture, but you can imagine that everything is jiggling around, moving around, and so on and so forth. And you can see at once that the cell here, whose sizes could be, let's say, 20 microns, houses many, many different kinds of membrane-bound compartments. Okay? And each of these compartments are given different names because they have different morphologies, they have different functions, they carry different sets of molecules. Okay? And so they're all given different names. And as I said, we're going to be looking at this extremely beautiful system of compartments called the Golgi apparatus. Okay? But we'll also talk about the other ones. But Golgi is going to be the focus of this. And all this is embedded in a very highly viscous cytoplasm. And crisscrossing this entire cell are a very dense meshwork of stiff polymers, actin and microtubules. Neil must have talked about that. But what is, of course, missing in this classification are these objects. For instance, lipid droplets, there are several lipid droplets floating around in the cytoplasm. Membrane contact sites. Every membrane is touching every other membrane, and there are very specific and functional membrane contact sites between organelles. And finally, something that has caught the fancy of people, of biologists and physicists across the world, the membraneless compartments, that is, organizations of chemicals within the cytoplasm, within the nucleoplasm, which house very specific large molecules, okay, such as ribosomes and so on. So that's, of course, these kinds of organelles or compartments are missing from this older picture. Okay. So now first I'll talk about the logic of compartment, why it all have compartments. And the common understanding is that these compartments are useful in sequestering molecules, very specific biomolecules. They're also useful in synthesis of molecules, including ATP, which is largely synthesized in the mitochondria, in one of the organelles that I talked about. Since they synthesize very specific molecules, they are crucibles for chemical reactions. So you can have independent chemical reactions going on in different parts of the cell. And because chemical reactions is the cell's way of doing information processing, this means that I have individual and independent signaling hubs or information processing units. Okay? It's good to think about chemistry as chemical reactions as an information processing unit. That is now given rise to an additional layer of understanding the logic of these chemical reactions. Okay. So Now, given that they are, these compartments which sequester molecules, engage in their own chemical reactions, clearly these have to talk to each other in some way, right? And the communication between different organelles within the cell are along established communication routes where they exchange information, they pass information amongst each other. And so this dynamical coupling between autonomous compartments, okay, immediately should evoke the idea of emergent distributed systems. Okay? So this is just like the language that is used for computers. You can have individual computing units which are doing their own autonomous calculations, but then they need to interact with each other. Okay? So again, what immediately should come out to you, just like what you have in computers, is what is the notion of time? Is there a master clock which tells individual computing elements, dictates a global time to all individual computing elements, or are there local clocks, right? And if they're local clocks, how do they act in a way so that there is no conflict? Yeah. So this is the famous problem of timestamps, logical timestamps in computers, and this kind of setup you should be asking questions such as that, to the cell. Okay. Now, so that was for the logic. Now let's just look at the physical characteristics of these functional compartments. All these compartments, except for these membraneless objects that I talked about, all these compartments are encased in a multi-component asymmetric phospholipid bilayer membrane. Okay, they're made up of lipids. This is a bilayer lipid. Since it's bilayer, it has two layers. The upper leaflet of that layer has lipids which may be different from the lower leaflet. Okay, and there are many different kinds of lipids. So that's what that means: multi-component asymmetric phospholipid bilayer membrane. Of course, on this membrane with lipids are embedded many proteins. Typically, the membrane thickness is dictated by the length of the lipids in the two layers, and that turns out to be varying. It varies in compartments. Okay, and it doesn't vary randomly. There's a systematic variation. So, for instance, this is the picture of the Golgi, and this is called the cis. This part is called the cis Golgi, and that's called the trans Golgi. The thickness of the membrane systematically increases as you go from cis to trans. Okay? And that's because the lipids get longer and longer. Yeah? Because there's a processing of these lipids which enzymatically keeps adding, increases the length of the lipid. Okay? Now this is an important thing. And why is it important? Because this turns out that if you have a protein which is embedded in the membrane of a given lipid, it likes to have a matching. The size of the protein that is embedded in the membrane typically likes to match with the lipid size. And so if there is a mismatch, then it is easy to come out and go to the next system. Okay? So this is a way to localize proteins within that compartment, localize membrane proteins within the compartment. Okay? Because so of course now what you can ask, what happens to the interior? What is contained in the interior of these compartments? What is contained in the interior of the compartments is of course an aqueous medium, which has many different kinds of proteins, enzymes, and carbohydrates. Okay. Now, surprisingly, the pH of this aqueous medium can also vary systematically. Okay? And this is another level of control that is happening in this. So, for instance, in the Golgi, the pH within the cell is largely acidic. That is, the pH has a lot of H plus ions floating around, and very systematically, the pH in the cis compartment has a higher value than the pH in the trans compartment. Okay, and this gradation or variation or gradient of pH is maintained actively by pumps. Okay? Yeah, there are pumps, protein pumps, which keep pumping in H plus ions, and it does it in a very with a feedback control, which maintains the pH of each of these compartments at the same value. Okay? So, okay. So as the bottom line of this is physical properties such as membrane thickness and pH can vary systematically across organelles. Okay, and this is going to be used by the cell to do interesting physical things. Yeah? Because this is almost like imposing a imposed gradient of something, right? Temperature gradients, for instance, will drive energy flow. Right? Chemical potential gradients will drive particle flow. Similarly, this is the cell's way of maintaining gradients. Right? You can't maintain gradients continuously because you have discrete compartments, but you do that by having uh, an identity, a physical identity, distinct physical identity for each compartment. Yeah? So it's a very clever way of doing things. Now, just to give you a picture of this, of the amount of material that is there inside. Okay, here I'm looking at area and volume. Okay, this is taken from Rob Phillips and Ron Milo's book, which every one of you should read. It's Cell Biology by Numbers. Okay, it tells you how to compute do a back of the envelope calculation for all kinds of numbers in biology. Okay, and so, for instance, what if you look at here? So these numbers are in percentage of total cell membrane. Okay, so if you look at the plasma membrane, this has in this particular cell, liver hepatocyte, has 2% of the total cell membrane. Okay? 2% is pretty small, right? Right. So most of the membrane is actually inside. Most of the membrane of the cell is actually inside the cell. Okay? For instance, here the endoplasmic reticulum has 35% of the total cell membrane. So clearly, if it has to be inside, it has to be totally ramified, right? 35% of the membrane of the total cell membrane is actually occupied by the rough ER, the rough endoplasmic reticulum. The Golgi, which is the organelle of that I'm going to talk about today, has 7% in liver hepatocyte. Okay, so you can see all these internal structures are very, very, very ramified. Okay, right, because it's being covered by a fairly non-ramified plasma membrane. Right. Similarly, one can do the same lookup. It's actually nice to look at these numbers. You know, it's important for a theoretical physicist to look at numbers and because the only way to come to a correct theory of whatever phenomena that you have is to look at these numbers, make theories, and then confront them with experiments. And what one hopes is that most often your theories are wrong, so that you have a better understanding. Okay? So, I'm sorry, I'm going to give you gyan like this, but so, okay. Okay. So that's about the sort of comparative cell biology of the organelles. Yeah. So what I'd like you to appreciate is that there is also a notion of time in cells. Right. At Longwell time scales, the time in cells are set by the cell cycle time. Right. So there's a constitutive time scale called where the usual happenings in the cell go on. That's called the interphase. But when the cell decides to divide, that period is called mitosis. And surprisingly, these compartments, both the shape, the form, and the size of these compartments vary as a function of the cell state, okay, as a function of its time. Okay. So at interphase, for instance, which is the normal constitutive pictures that people show, this is what the Golgi, remember the Golgi was looking like that, right? Okay, there's these pancake-like structures. But when you're close to the cell division, then they get this Golgi gets fragmented into tiny vesicles. Okay, and that's shown here. This is the interface configuration of interface, which is the constitutive cell state, the configuration of the Golgi, and at cell division it breaks up here. Okay. Similarly, if you look at the mitochondria, which is the organelle that synthesizes ATP. Okay. Again, it has a tubular structure at interface, shown here. Okay, but during mitosis, during cell division, it breaks up into tiny fragments. Okay, so it's interesting that the organelle, shape is not only governed by local physics, right? But is actually talking to what is happening in the cell. So there has to be some communication, some signaling, right? Which one has to then have a theory for. Okay, this is nice. I just wanted to show this experiment where this is done by Nagaraj Balasubramaniam's group in IISc, Pune, where you look at a cell adhered, a fibroblast that's adhered onto a substrate, and look at the Golgi, and it looks like the interface. It's just like that. Okay, there are certain markers, molecular labels that have been placed on this to make you visualize it. But now, when you detach the cell from the substrate, so that's suspended out there, right? The Golgi fragments into these tiny bits, right? That's remarkable, isn't it? I mean, I find this remarkable, and this is a recent paper that shows this. So the shape of the Golgi and presumably many other organelles depends on the environment that the cell is embedded. Okay? So not only on the state but on the environment the cell is embedded. Okay. Right. So this is what I just said. That so now given that for instance in the interface in the normal constitutive form, namely interface, the Golgi looks something like this: well-stacked pancake-like structures between the endoplasmic reticulum and the plasma membrane. And we also saw in the last slide that the Golgi fragments during mitosis. Clearly, when the cell goes from mitotic state to the interface state, this kind of configuration, this kind of fragmented configuration, has to self-assemble, okay, to form this, right? And that's an interesting question to ask in terms of physics, that given this as the initial condition, given the rules of non-equilibrium self-assembly, how do you go from there to there? Yeah? Okay, so, right. Okay, so everything in all the organelles that I talked to you about are dynamic, and how do I? Yeah, okay, they're all dynamic. What you see here is the endoplasmic reticulum, which is a tubular network, which by very recent microscopy techniques have been shown to have these tubules, connected tubules, very tense taut tubules, which are fluctuating in a very active manner. Okay, this is the endoplasmic reticulum, as I said, presumably fluctuating because of motors which pull it and push it, and there's a turnover of motors and so on and so forth. Okay, this is the Golgi, where again, as you saw, we'll come back again. So what you see here is a labeled protein called VSVG. It's a small protein, and that protein fluxes through the Golgi, and by attaching a fluorophore to that, you can sort of visualize the Golgi, and you can see that it's very dynamic. What you see in red is our microtubules, and these vesicles that come out from this are moving along microtubules. Okay? But the interesting thing is that this Golgi here is actually, by higher resolution microscopy, seen as these distinct compartments, is maintained, okay, in the presence of this flux of tiny cargo. Okay, so this is a non-equilibrium steady state with a constant current. Yeah. So those of you who have studied non-equilibrium statistical mechanics, this is a beautiful example of that. Okay. This is the mitochondria, which undergoes, again, tubular structures which undergo fission and fusion. And so at the end of the day, they'll have a distribution, a steady state distribution of tubular sizes, okay, which in principle can be measured, right? But this steady state distribution is distinct from this. This steady state distribution does not carry a constant current. There's a fixed amount of total mass in the system, okay? Whereas here, there's a constant rate at which material fluxes. Okay? So there are two different ensembles of, if you want to pose it as a statistical mechanics problem. Okay, so here I'm talking about dynamics at steady state, and what I talked about earlier was dynamics of the assembly. Okay, this is the dynamics of the assembly. So these are kinds of questions that we'll be asking. Okay, now these organelles, as I showed in the other picture, are all the cell is really packed. So the organelles are really touching each other. They're not only touching each other, they're also touching the cytoskeleton, which are these stiff polymers of actin and microtubules. And so you can ask whether how strong is the organelle-cytoskeletal interaction? And what does the cytoskeleton do to the organelles? Yeah? And as you see here, organelles facilitate the trafficking of vesicles, but they can also provide a scaffold, okay, by adhesion or entrapment, and therefore change the shape of these organelles. Okay? So that's the other thing, and it can also then give rise to precise spatial locations. It's surprising that inside the cell is supposed to be a fluid, right? Highly viscous fluid, but still the organelles are sitting where they are. Even when the cell is moving, yeah, the things are not getting mixed up. No, even if you take the cell and rotate it, the things won't get mixed up. I mean, of course, as you rotate it really, really fast, then that will happen. But if you rotate it at reasonable speeds, it doesn't get mixed up. So it's somehow being held at a very precise place, right? And that's I mean, in terms of self-organization, it's really remarkable to think about that, right? And so this provides not only a scaffolding but also precise cellular geography. Okay? And how does that come to be is an interesting thing. More recently, like here, this is the paper from Nagaraj Balasubramanian's lab, which shows that upon suspension, that is upon detachment of the cell away from the substrate, there is a total change not only in the shape of these and the volume occupied by the Golgi, but also they are geographically totally misplaced from the wild type. Okay, and that has to do with the fact that upon suspension, cells sort of lose their stress fibers. Okay? The actin is much more fragmented and loses. You don't get these stiff actin filaments there. Okay. More recently, in collaboration with Vaishnavi in Australia, currently in Australia, we've been looking at the coupling, the interplay between organelles and microtubules in fission yeast. And what we find is that a precise partitioning of mitochondria during cell division of fission yeast is contingent on the fact that the mitochondria, that the microtubules are coupled with the mitochondria. So in the absence of that coupling, or if you change the size of the mitochondria or the microtubules, this partitioning doesn't happen. So it's remarkable that in every instant there is this, biology makes use of whatever available, right, to ensure high fidelity of very important functions. Okay? In this particular case, you better partition the mitochondria almost equally, else there'll be problems in further generations. Diffusion is not enough. It's a very active process, but it's also a very precise process. Though actually, there's been only one set of papers on this by Rafalski and Johan Paulsen, where they tried to analyze the precision of partitioning across generations, and whether the cell needs to have a control mechanism to ensure that if there are errors. So, but it's still an open field to study. Okay, and so that was about organelle cytoskeletal interactions, and I just wanted to flash a recent 3D super resolution reconstruction from Genelia farm, from the Genelia labs, which shows how these organelles are touching each other. All these organelles that are listed here are touching each other. Okay? And so there are clearly contacts between organelles, which have a functional role. It turns out that the mitochondrial division, fission and fusion, is contingent on the contact between the mitochondria and the ER. Okay, who would know this? But that's and then there are these places where the ER touches the. Remember, the ER is a very ramified, high area structure. So oftentimes it touches the plasma membrane, and those touching sites, they're called ER contact sites. They are highly specialized, and they allow fluxes of material to go onto the other thing. it's quite remarkable. You know, people have recently introduced molecules, proteins, labeled proteins from the outside into the cell. And they've tried to ask, once I introduce them on the membrane of the cell, how fast does it go inside? And it zoops through. It goes really fast compared to diffusion. And one of the ideas is that maybe it's seeking these ER exit sites, these ER contact sites, and then moving along the ER. So yeah, but phenomenology in biology never ceases to amaze one. And it's important in order to do biology and understand these, because there's no way, I mean, though theorists are only limited by their imagination, I think theorists are nothing, that the imagination of theorists is nothing compared to the avenues explored by evolution. So I think it's very important for all of you to look at phenomenology and address physics ideas and physics questions to this area. Okay, and finally, I think I have 2 more slides on this. This is part of the phenomenology. Finally, I'm going to talk about this. Well, I won't talk about this, but I think an important question is, how does the size and number of compartments scale with cell size? Okay, when the cell becomes bigger, do the compartments also become bigger? Do the number of compartments change? So on and so forth. It turns out that these are extremely challenging, new, and difficult questions. Okay? So this was an example that where one shows that the size of the nucleus, the size of the nucleus scales with the cell size. Okay? And this is the experiments which show that. What the experimentalists have done is to inject a small nucleus in a larger cell, which has a larger nucleus, and over time, the small nucleus, so the cell then becomes, when it becomes bigger, you find that that introduced nucleus also increases in size, and the other nucleus sort of decreases in size. And why is that? And that's because both of them are competing for the same pool of material that is found. Okay? So this is evidence of something called a limiting pool mechanism, saying that the constitutive ingredients that make up the nucleus are fixed. Okay? And when you have a larger cell, you have more of this constitutive nucleus, constitutive ingredients, and so you build a bigger nucleus. If you have the same size of cell and you put 2 competing nuclei in it, then both of them are accessing the same pool. So you'll see that one grows at the expense of the other. Okay. Okay. Yeah. Okay. So now, having said all that, we'll now go to fission and fusion. Okay. Yeah. How am I doing with time?
Let's say half an hour.
45 minutes. Yeah. Thanks. So yeah, I've been going slow. Sorry. Okay. So now we'll talk about this process, or how do I do that? No. Oh, this didn't work. Okay. Yeah, we're going to talk about this, namely the non-equilibrium dynamics of the Golgi. The Golgi is here. The Golgi is a system of compartments called cisterna. They're typically pancake-shaped and very highly ramified structures, and they lie between the donor ER membrane and the plasma membrane, which is the cell surface. And what is happening is that tiny, tiny vesicles, as you're seeing here, tiny vesicles come out from the ER and fuse with these cisternae, and then they fission from the cisternae. Then, from the cisternae, little tiny vesicles fission out. They travel a little distance, fuse again, and so on and so forth. So there is a sequential and stochastic fission and fusion process that maintains this steady-state architecture. Okay, and what we're going to ask is, despite this stochastic fission and fusion process, is there a steady-state size of this? Is there a steady-state shape? how is the shape, what are the fluctuations of the shape, how is the shape maintained despite this flux of material going in and out. Okay? Remember, again, I wanted to stress this, that these are non-equilibrium, this is a non-equilibrium system. We're asking questions about the non-equilibrium steady state of this structure in the presence of a flux of material. Okay. Now, what is fission and fusion? Fission and fusion of little tiny membranes onto this mother membrane is a process that is an active process, that is an energy consuming process. Okay? And it consumes energy through these GTPases, okay, which are just GTP is just like ATP, and with a different nucleotide, and it's this chemical cycle, a non-equilibrium chemical cycle, which involves the flux of the constant flux of energy that drives the fission and fusion process. Okay, so you can see here that I'm going to bring in active physics and active mechanics into the process because of this non-equilibrium chemistry that is going on at different points, at every point on this membrane. Okay, and at the end of the day, this non-equilibrium cycle is going to drive the fission or fusion of material. Okay. Yeah. Now, because it's a non-equilibrium chemical cycle, this non-equilibrium chemical cycle drives the current. Okay, and this current here, this is a probability current. This current here transduces it into fluxes of molecules and forces. Yeah. Yeah. Okay. So, for instance, here you can represent the chemical cycle by this Markov four-state Markov process with transition probabilities that go from 1 to 2 and back. You can see that the arrow here is longer than the arrow there, which means the transition probability from 1 to 2 is not the same as the transition probability from 2 to 1. Okay? This mismatch or this asymmetry or imbalance between transition rates is what violates a principle called detailed balance, which usually is invoked in equilibrium systems. Okay? So, so it's because of this imbalance of the transition probabilities that you can systematically drive a current through this closed chemical cycle. Okay? And this current here that is driven through the chemical cycle is actually maintained by holding the levels of ATP and ADP fixed in the cell. Okay? It's like having a pressure gradient which drives a fluid flow. Here you're having a gradient of chemical potential between ATP and ADP, which drives the current here in this chemical cycle. It drives the chemical flow. Okay? And associated with this current can be now transduced into forces. Typical forces for myosin, for instance, are these 14 pascals. Okay. Now, yeah. So, has there been any lecture on detailed balance and so on? Yeah. Yes. So I can skip this, or yeah. Okay. Okay. So, but I just wanted to put one point here. Maybe I won't skip it because I need some of these things. Okay. Have you done Kolmogorov loop condition? You're done. Oh, okay. Then I don't need to do that. Okay. Good. Okay. So then I can skip all this and go directly here. Okay. So you know that there is a necessary and sufficient condition which ensures ensures the violation of detailed balance. That is, if Kolmogorov loop condition is not satisfied, then in any loop that you can find in your network, then that network, the chemical network or the Markov process, will be driven out of equilibrium. Okay, and in general carry a current. Okay, so now let's now look at the mesoscale physics of fission and fusion. So let's say fusion here involves the apposition of two bilayer membranes coming together. Okay, where then as you press them together and you'll generate membrane stresses, the membrane stresses can be relieved by this process of fusion. Okay? And the way fusion proceeds was predicted by theory, principally from Misha Kozlov, to show that there is an intermediate stock region, then there's a hemifusion diaphragm that forms, and then finally relieving the stresses by this fusion pore. Okay? So in doing so, it has to cross a free energy barrier. And a lot of numerical studies and indeed theoretical studies have been used to estimate this free energy barrier. Okay? What is the energy required in order to fuse membranes together? Okay. It turns out that simple models of elastic models of the membrane, which Neer might have talked about, Helfrich models of the membrane. Oh, you haven't? Okay, the next. Okay, yeah. They overestimate the barrier-free energy, but if you include things like lipid tilt, so some liquid crystal physics, then you sort of estimate that this sort of gives results that is consistent with numerics and with experiments. Okay, so these are experiments which, for instance, see this hemifusion, the fusion pore at the end of the fusion process. And these are all atom simulations, which also reveal the presence of the stock expansion, hemifusion diaphragm, and fusion pore. And the energy scales that I've written down here, the free energy barrier to come to this state, that is of the hemifusion diaphragm, is around 40 kT, 40 times room temperature, whereas the energy cost in going all the way to the fusion pore is around 100 room temperature. Okay? So you require that much energy for it to. So clearly, it can't be a passive process. Yeah, you need to. So it's not to say that passively membranes can't fuse. If you have, charged lipids, you could have a positively charged lipid and a negatively charged lipid, and then they could come together and fuse spontaneously. Okay, and there it, of course, derives this energy from the electrostatic energy. But these membranes are not charged so heavily, and so it requires local energy inputs, active processes to drive this fusion or fission process to completion. Okay, and so these are the many proteins. This is one of the humongous number of proteins that is involved in the fusion. This is called snares, and it's the elastic energy that is obtained that is sort of stored in the twisting of these braiding of these snares that drives membranes coming together and ultimately fusing. Okay? Yeah. So typically, this is a cycle. This is a fusion cycle where the vesicle here tries to dock on this mother membrane and then undergoes a fusion cycle, where at the completion of the cycle, this vesicle has merged with their mother membrane. Okay? So that's a process of fusion. On the way, of course, it involves a lot of active processes, such as coming from calcium influx, energy from ATP, and so on and so forth. Okay? Remember, it has to cross that free energy barrier of 100 kT. Right? So it needs all this. This is the equivalent in the fission process, where again, the mother membrane here gives rise to a local bump, which is covered by something called coat proteins. And these coat proteins then finally result in a scission of this vesicle away from the mother membrane, which involves the hydrolysis, the phosphorylation of GTP. Okay. Now, before so now you see that the fusion and fission processes are active processes, okay, and have to be treated via the rules of stochastic thermodynamics or active mechanics. And this is what I'll be talking about in the subsequent lectures. But before we go to the next, the kinetics. So that's the second part of that. I wanted you to bear in mind two conventional models. There are actually more models, but these are the most sort of celebrated and quoted models in the organization and dynamics of the Golgi. So this was my schematic cartoon of the Golgi, right? This is the endoplasmic reticulum, a source of material of these tiny vesicles, which go and fuse via processes of fission and fusion, form ultimately at steady state these, let's say, three different cisterna, Golgi cisterna, okay? And that's the plasma membrane. But in point of, oops, that didn't happen. But actually the whole process is quite dynamic. And what is actually happening is that, okay? One theory called the vesicle transport theory suggests that these cisterna are actually fixed in space. Okay? And tiny vesicles emanate from the ER and then fuse there, fission out, fuse, etc., etc. Okay? So this is called the vesicle transport model. A more recent model and a more clever model is the following, and this is called cisternal progression model, where this compartment, see, has formed de novo, like that. Yeah, has formed de novo from the emergence of tiny vesicles from here, which fuse to form that. Okay, and then these cisterna then move and is replaced by another one, and so on and so forth. So what was initially a cis has now become a trans, and so on and so forth. Okay, so this is like a treadmilling, of these things, and this has been called the cisternal progression, a cisternal maturation model, and there's copious amounts of controversy between the followers of this model and that model. Of course, there are models in between. There are many, many different kinds of models. As yet, not extremely well resolved, but I think people agree that in mammalian cells, this is the kind of model that most correctly describes the dynamics of the Golgi, whereas in budding yeast, for instance, there is evidence that this is the appropriate model. Okay? So again, there's nothing universal about biology. It could be depend on, right? Because evolution does its role in different places independently. Okay. So now, so I'd like just to keep that in mind. A question that you should ask in this case is in that model, you saw that the oops, sorry. you saw that the cisternae are moving, right? And so the question that as a physicist you should ask is, what is the driving force for movement? Okay? Because this is clearly an active process, right? And one should try to ask and resolve the question of what is the driving force. And that indeed is the question that I'm going to discuss in the next lecture. Okay, so what we'll talk about today is kinetics, not mechanics, as opposed to mechanics. And we'll be talking about the kinetics of non-equilibrium assembly of a system of Golgi system. Okay, there's many stacks here. This kinetics of non-equilibrium self-assembly, as I described earlier, involves the influx of material from the ER. It involves fission and fusion. It involves transport of tiny vesicles. It involves chemical changes. As you can see here, there are different colors, which says that the chemistry here is different. The chemical composition here is different from the chemical composition there. Okay, and it involves the movement of the entire cisterna, as you saw in the cisternal progression model. So these would be the dynamical rules, the kinetic rules that you would use to describe the kinetics of this self-assembly. Okay, but before that, as any physicist should do, you have to look for numbers. And for getting numbers, you have to then look at the experiments, analyze them, and extract these numbers, these kinetic parameters. Okay? And so this is what Serge Dmitriev did, where he looks at existing experiments at that time. Okay, the experiments that existed at that time, and reanalyzed all those experiments, but from a different, from a physicist's perspective, in order to extract kinetic parameters. Okay, I won't maybe go through the details of this, but suffice to say that these are, so for instance, one simple experiment could be, and I'll just describe one experiment. One simple experiment could be that I block the exit of these tiny vesicles from the ER. Okay, somehow. You can do that by doing a temperature shift. Yeah? And then I had initially, there was a lot of material that was here. Okay, I label those materials with the fluorescence dye, and I see how does this material that is here decay in time, because it's no longer getting material from the ER because you blocked it, but material can go from this region to the plasma membrane. So it keeps losing material, and that will decrease the intensity of light, of the fluorescent light that you see, right? So these are curves such as that, which show the decay of that. And one can now have a theory to describe this. The theory will involve all the parameters that are there in these rates, and and then we can try to extract kinetic parameters. So here is the theory that you can have. Cn is the concentration of this labeled protein in cisterna n at time t. Okay? And you can now write down a sort of difference equation for how this concentration evolves in cisternal index, because it transfers material from one cisterna to the other, either forward or backward. Okay? And as a function of cisternal index and time. So the terms in the equation, which you can derive from more microscopic theory, is a diffusive term, like so. Okay, so this is where dt is the effective diffusion coefficient of this material, Cn. Okay, and then there is an advection term. The advection term has two parts. One is the difference between the forward hopping rate and the backward hopping rate, k minus k prime. Okay? And the other term is the net translation of the cisterna as it moves from the ER, from the side of the ER to the plasma membrane. Okay? So this is a net translation, which is a combination of those two. And then there's possible leakage. After all, material can go from there to some other organelle. Okay? So this is the kind of effective theory one can derive, and one can now interrogate the experiments by this equation and try to extract these various rates that you see here. Okay? So here's what you, at the end of the day, after doing those comparisons, you find the following time scales. Yeah? You find that the average transit time, that is the time it takes for material to go from the ER to the plasma membrane, is around 15 to 20 minutes. Okay? In 15 to 20 minutes, a typical protein will traverse that distance. Now, it turns out that the diffusion coefficient here is extracted from this analysis is 0.2 per minute. Okay? Now, what does that mean? That means the following. That means that every cargo, every protein that you're labeling, okay, be it small or large, because the experiment was done on both small molecules and large molecules. Large molecules like procollagen are diffusing, which means it's going back and forth. Right? And that's interesting because people had until that time thought that large molecules only go forward. They don't come backwards. But this one says that even large molecules will come backwards. Okay? So while there is a net vectorial flux from the ER to the plasma membrane, on an average, things are moving forward. At a micro level, things are moving back, like so. Okay? Okay. Further, what we found is that the experiments do not do not say that the cisternal progression rate, that is the movement of the entire cisterna, is 0, but is actually bounded. We can bound it by this. It could be 0, but the experiments done so far are not sharp enough to lower this bound. Okay? So at the end of the day, this analysis said, it is a mixture of these two models. Okay? A pure cisternal progression model does not work. Okay? And you can't establish for sure that is a pure vesicle transport model. What is most likely true is a combination of these two. Okay, so, but more than that, I think it gives you numbers which can be now used. Okay. Now I just wanted to state here that it's important to have the right level of abstraction. Okay, when you're dealing with biology. If for instance in our analysis we had been too coarse-grained, then you would have said, oh, this is like a biased random walk, right? And you would not have extracted anything of biological interest. You would have been happy, okay, that your analysis fits the data, but you won't learn anything from it. Okay? Because indeed it is a biased random walk, but that's a wrong level of coarse graining. It's too coarse-grained. Okay? Further, if you're too detailed, then you lose the ability to to generate abstract organizing principles, okay? Abstract general organizing principles. And you get totally overwhelmed by molecular details. Indeed, the first analysis of this kind of data done in Jennifer Lippincott-Schwartz's group used a simulation model which involved 30 parameters. 30 parameters to fit that. Okay, so this is really an overkill, and of course it doesn't establish anything, and so that's not the way one should do the analysis. Okay, yeah, so that's just a point I'd like to mention. Okay, so here is what we want to do at the end of the day. I mean, end of these three lectures, and in fact, in further work that I'm hoping to do, is this is the kind of questions we want. We have now a system. We have numbers now obtained from kinetic these experiments, and now using these numbers, hopefully, we want to be able to address the following question. Here we have a system of cellular compartments. They are embedded in a trafficking pathway. They are embedded in a highway, which involves a lot of trafficking back and forth, okay, from the ER to the plasma membrane. What I want to be able to do is to find out the steady state distribution of the size, the shape, the number of compartments, the composition, the chemical composition in the presence of a finite current. Okay, this is the non-equilibrium stat-mech problem I want to address. Okay, and in the process, on the route of doing such a thing, I want to be able to discover how does the size control of the Golgi happen, despite the fact that fluxes are coming in and out and so on and so forth, and what maintains the relative spacing between these compartments. Okay, what is the cellular geography? Okay, I'm not saying that we're going to address all these questions, but that's the hope. We'll try to get there. Okay, so here's the start. Again, how much time? 5 minutes more? Okay. Oh, you had a question? Sorry. Please.
In the analysis you have done until now,
Yeah.
so in that I don't see this fusion-fission events, they are not incorporated in that equation.
In that equation, yeah. So remember I said that this is an effective equation that can be derived from a more microscopic theory. So the more microscopic has fission and fusion, has kinetic rates. And D here is the effective diffusion coefficient coming from fission and fusion. So that's the rate.
Fusion will have like a many body term, like C and C.
No, so this one. No, so remember it's a diffusion of tiny vesicles or this.
So this is just focusing on them.
Yeah, only on them. Right, exactly. Yeah. So
there is also like boundary condition on the part.
correct, exactly. Yeah. So there's a finite rate at which. I have 20 minutes. Okay. Yes. Okay. Yeah. Okay. So so we'll start off with some crude models and then make it more elaborate. Okay. So here I'm going to talk about a minimum non-equilibrium self-assembly with spatial order, which gets you the spatial order, or at least gets you some attempt at trying to get a spatial order. And this will be kinetics. It won't be mechanics, and it will involve kinetics and chemical processing. Okay. And the kinetics is going to be transport, fission, fusion, and here is going to be chemical reactions. Yeah. Okay. So here's the kind of cartoon that you have. You have a certain rate K in, which tells you the influx of tiny vesicles A, okay, with chemical composition A that comes out from the ER. Okay. They then fuse at a certain given rate, and they can fuse to form a structure. Okay. This simultaneously, or as this time progresses, this A here, this chemical A, which was only only decorating these tiny vesicles, and now eventually forming this, transforms to B. Okay, and then later on B transforms to C and so on, with some enzymatic Michaelis-Menten enzyme kinetics, with rates given by EA, EB, etc. Right. So that's the chemistry process, which is being overlaid on the transport process, transport fission fusion of the vesicles. Okay. Now, what we have done is to say that in general, the fission rates and the fusion rates, here we have only taken the fission rate, depends on the amount of, let's say for this, the fission of B vesicles depends on the amount of B vesicles in the cisterna. Okay? If there are, and it has this, roughly this form. It saturates for large MB, and it's an increasing function for small MB. And this is easy to see because imagine I have a, remember each of these fusion and fission events are orchestrated by some proteins. The proteins are limiting, right? There's a fixed number of these proteins there, right? So clearly for very, very large M, yeah, when M is much larger than the number of proteins that is available, the proteins are limiting, so it won't depend on M. Okay, and clearly this has to go up from 0. So it is typically of this form. Yeah. So similarly, if it is a Michaelis-Menten kinetics, then you could argue that the rate of chemical reactions, namely EA, EB, etc., has this kind of chemical kernel. Okay? It depends on the mass of the cisterna, okay, masses of any aggregate in this sort of hill kind of form. Okay, so more precisely, this is what we do. We consider a lattice model, but that's not crucial. You can also take the continuum limit. You start off with a source, namely the ER. Little tiny vesicles of unit mass get into the system at rate given by A, and then they hop from one lattice site to the other. Okay? There's a hopping rate to the right, there's a hopping rate to the left. There's a net vectorial of transport. Therefore, the asymmetry between the right movement and the left movement is represented by a parameter gamma. This gamma, which I'm going to come here. Okay. But then, when vesicles come together, they will fuse. Okay. Let's say with rate 1. Okay. But when vesicles fission out from, let's say, a compartment like this, okay, then it will happen at a rate given by some parameter WA, which we are going to vary, multiplied by this fission kernel, which has this form. Yeah, so it's a mass-dependent fission kernel. Okay. Now, further, to incorporate the notion of cisternal progression, I'm going to have with rate D, I'm going to have these bigger cisterna move, okay, stochastically move in the net this direction, in the net direction to the right, towards the plasma. Okay. In principle, the fission can carry vesicles either to the left or to the right, and as I said, the asymmetry to establish net vectorial flux is represented by gamma. Okay. So is that clear? You have, and on top of that, you have chemical reactions. A going to B going to C with rates U, B, etc. Yeah. So this is clear. You have transport, fission, fusion, chemical reactions. Yeah. And then what you want to do is you want to write down a master equation for what is the mass, okay, of an aggregate at site I as a function of time. Yeah. But mass of A kind of things, or mass of B kind of things. Okay. Is that clear? This is silence. Yeah. Right. No, it could happen in the vesicles too. So for instance, this vesicle could certainly turn. So that, yeah, can do. So that's of course not what happens in the biology. Well, these enzymes are supposed to be housed in cisterna, but so we have simplified the chemistry considerably. It could be doing that, and so it could, yeah, that's true. But I don't want to sell this more than what it is. So I think it's very simplified, but it gives interesting features. That's why. Yeah. No, so you're doing this. So we're going to write a master equation for which we're going to do a Monte Carlo simulation, and you could do that. You could do simultaneous updates or single updates or whatever. You can go to a site. You can choose a site randomly and then do it. Is that what you're saying? Yeah, 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 yeah. It's site dependent, right, exactly. As opposed, that's a good point. So as opposed to this calculation, which was cisternal dependent, this one is site dependent. Yeah, that's a good point. Okay, so now we can write down the equations, kinetic equations for how the mass at site I of A type changes with time. Okay, and it'll change with time because it's getting material by fusion, it's removing material by fission. So that is these terms. Okay, and I said this fission kernel depends on the mass. Okay, and then there's chemical transformation. Okay, with rates u. Remember, so A goes to B with rate u, B goes to C with rate B, etc. Okay, so this is the fission part of the master equation. This is the asymmetric transport. It could hop to the right or go to the left, and this is the chemical transformation. Yeah. Okay. Now you can of course write down. This is the equation for MIA. That is the mass of A type of objects at site I. Okay, I could also do this for mass B, for the color B, chemical B, and chemical C. Okay, so I have, depending on the number of flavors or colors, number of colors that I incorporate, I will have that many equations, kinetic equations. Yeah, and at each site, to get the total aggregate, I have to sum over MIA plus MIB plus MIC. That gives you the total mass, total size of the aggregate at site I. Yeah. Okay. So a typical, yeah. Okay. So a typical thing will have, there'll be the ER. Let's say there is the ER, and then that's the plasma membrane scaled to have a distance 1. And typically, what you'll find is that nothing, or very little mass in the intervening places, and then suddenly a large mass. Then nothing, then a slightly different mass, and so on. Okay, and so those things in steady state will tell us that these are aggregates. So, for instance, if I don't have cisternal drift, that is, I don't have cisterna moving with that rate, right, like in cisternal progression, then what I get in steady state are configurations like this. This is what happens in steady state. This is the color here denotes the A aggregate. This is the B aggregate, and that is the C aggregate. So you can see that you are recovering, okay, vesicle transport-like phenomena. Okay? These are aggregates that have been built up by the microscopic kinetic processes that I told you about. And in steady state, there's still a flux of material going through. Okay? These are not, it's not that this is static, right? The aggregates are static, but the little tiny material is still fluxing back and forth. Okay? So this is a non-equilibrium steady state maintained in the presence of a flux of material. Okay? Now, okay. So what is this for? Yeah. Okay. So what this says is that you have distinct cisternae. Okay, distinct cisternae, both morphologically and chemically. Okay, and now you can ask what sets the scale between the distance scale between these cisternae, and it turns out that the distance scale is set by the ratio of the chemical rate. Remember U, V, etc., chemical rate by fission rate. So here is an interesting thing, right? The distance, at least within this model, I'm not saying that this is reality. Within this model, distance is set by a ratio of time scales. Okay, I find that interesting. It's not that there's a length scale in this problem. I mean, there's a length scale, of course, lattice spacing, but that's the only length scale in the problem. The rest of the things are just rates. Okay, and so this is saying actually that the chemical reaction rates and fission rates are actually dictating what is the distance between these cisterna within this model. Now, of course, in reality, there could be cytoskeleton, this, etc., which dictates it. We haven't incorporated any of that. But here is a testable hypothesis in principle. If you can extract the cytoskeleton in some way, this is a testable thing. Okay. Okay. What if you allow for drift to happen? What if you allow for cisterna to move? Okay. What happens is that this forms a steady state. The steady state is much more interesting. The steady state is no longer these three distinct compartments of A, B, and C, but actually has this kind of feature, where if you fix yourself at lattice position or spatial position X, then you have something of C, something of B, and something of A, which means you have cisternae which are mixed composition, right? And this is what people have seen. Nakano and Ben Glick have seen in budding yeast. They zoom into a little cisterna, Golgi cisterna here, okay, and then they see that multiple colors corresponding to multiple chemical composition. Okay, so indeed, by allowing for cisternal drift, it recapitulates the chemical features observed in cisternal maturation. Okay, budding yeast is the prototypical example where cisternal maturation or cisternal progression is supposed to work. Okay. Further, in the steady state in the cisternal, when you have drift, is no longer static in time, but is actually very dynamic. Okay, and so what is once this kind of pattern suddenly changes, statistically the same, but suddenly changes in position. Okay, it turns out that this is a very peculiar steady state because it's associated with very giant fluctuations. Okay, system spanning fluctuations, but that's not what you see in biology. This is of interest in physics. Okay, and what happens is that, so what this is saying is that if you look at a single cisterna, then you should see a time-dependent change in the composition. Okay, and this is indeed from the same set of people, Nakano's group and Ben Glick's group. They show this is the sort of temporal profile of these enzymes that are housed in these cisternae. Okay? And you can see that they change in time. Okay, so finally, yeah. So this I might actually finish in time. Okay, so okay. So now, so that's as far as we addressed the notion of spatial organization and structure formation. Yeah, but we haven't really looked at the details of size control, and that's because the model that we have was a bit too crude. It didn't put in the process. There was no mechanics, right? There's only kinetics, and the process of fission and fusion was a bit elementary. Yeah, we need to put in more physics of that. Not too much physics, but at least more of the physics. And the important element of the physics is the following: is in the rate of fission and fusion as a function of organelle size. Now you will appreciate, and this might have been talked about by Neer, that if the fission assembly rate and disassembly rate, namely fusion rate and fission rate, are independent of the organelle size, that is, they are constant, then you'll never get a finite structure. Okay, because if the fission rate is more than the fusion rate, it'll break up. If the fusion rate is more than the fission rate, it'll grow and expand to infinity. Right? So you need to have the fission rate, which is in this gray, intersect with the fusion rate, which is in red. Yeah. So that intersection point here corresponds to a stable fixed point. Yeah. So beyond this point, when the size is beyond this point, then disassembly is greater than assembly, so you decrease. Below this point, assembly is greater than disassembly, so you increase. Okay, so you make that a stable point. So that's what you have to achieve. This has been noticed in many in cytoskeletal structures, etc., but also happens in membranes. Yeah, the important thing that we need to do is to see if we can provide a microscopic mechanism by which you can achieve this. Right? And that's what I'm going to talk about here. So what we need now is a feedback control on fission and fusion, so that you get this kind of scenario. The fission here should be greater than the fusion at large M, but should be less than, the fission should be less than fusion at small m. Okay? And it should have this form. Okay? So this is, so now, in what I'm going to talk to you about, implicitly, I'm going to assume that there are sort of cisternal nucleation centers. And this is one of the failings of the theory that I'm going to talk about, because I don't know whether there exist nucleation centers for the Golgi. Yeah. So, but that's the hidden assumption in the model I'm going to present. Okay. So now here I want to remind you again of the fusion cycle, the fission cycle. So the fission and fusion involves again a non-equilibrium mechanochemical cycle that transports membrane area and membrane volume. Remember, tiny vesicle has both membrane area and volume inside. Okay. So when it fuses or fissions out, it's going to put in or take out both area and volume. Yeah. Tomorrow I'm going to show that it will not only take out area, volume, but also transfer momentum. Okay. So it can apply a force. Yeah. That's because that's going to be the subject of mechanics. But today I'm going to talk only about kinetics. Okay. And this process of fission and fusion involves these mechanochemical cycle. That is, involves proteins that talk to the elasticity or deformability of these membranes. Okay. So here is a simple four-state cycle for the fusion and fission. Okay. Why four-state? Because that's the simplest thing we can do. It has to be more than two in order to drive a current. So I'm going to take a four-state cycle. Yeah, and the four-state cycle, some of these nodes, that is, so I have a tiny vesicle. It's coming close to this to the compartment or cisterna. That's there. Okay, and when it comes close, it docks onto the membrane. That is state one. So these are going to be labeled the internal states of the vesicle. Yeah. So this is the docked membrane, docked vesicle. Then GTP is being consumed to go transition from state 1 to 2. Of course, there's a forward rate and a backward rate. That's like so. Okay, then it goes from state 2 to state 3 to state 4. Once it comes to state 4, it will fuse. Okay, so at the end of the cycle, of this non-equilibrium cycle, which drives the current through it, as we said earlier, okay, that will then result in a fusion process. Similarly, there's a corresponding thing on the fission. Okay, and these are the chemical steps that are involved in each of these transitions. Okay? Okay. So now it's this chemistry that needs to be overlaid on the rate of change of the mass of a cisterna. Remember, the mass of the cisterna only increases when it reaches state 4, and here it only decreases when it reaches state 4. Okay? Okay. So, okay. I also want to remind you of two different ensembles. Constitutively, in interface, this is the correct ensemble to look at, namely a constant vesicle flux coming from the ER. At post-mitosis, this is the correct, limiting pool mechanism might be the correct ensemble. I'm not going to be talking about this now, I'll be only talking about this. Okay, so again, very quickly, one can write down as before a master equation, okay? And one has to be slightly careful here because it involves both the transfer of mass and the transitions from across the internal states, okay? So here what we're going to do is that we're going to first define a probability that a given cisterna has mass m, okay? And is associated with vesicles Vi, which is the vesicle at state I, okay? Remember, the state was given by these internal states 1, 2, 3, 4. Okay, so the vesicle could be either in state 1, 2, 3, 4. Okay, so you got a mass of a cisterna, you got vesicle at state I. NV is the number of vesicles that are available to the cisterna at time t, so that they can fuse and go into the cisterna. NE is the number of fission and fusion proteins that are available at time t. Okay, so they can use it to do fusion and fission process. So what I want to do is to write down an equation for the probability of a cisterna having size m or mass m associated with a vesicle in state I given at time t, given that I have NV and NE numbers of vesicles and fission fusion proteins available at time t. Okay, so that's the equation I want to write down. And so I need to, so these set of terms here, I won't go through the details, but these set of terms here, which involves fission and fusion, okay, are mass changing terms. Remember, the mass can only change when you go to the fourth state. Okay, these are mass changing terms. These changes do not involve any change in mass, but involve changes in the internal cycle, going from 1 to 2 to 3. Yeah? Okay. So these are internal dynamics alone. Okay. So now what we're going to do is we're going to use an adiabatic approximation. That is, in the adiabatic approximation, the internal state dynamics is very fast. Yeah? And so I integrate over that to get an effective dynamics of the changes in the mass. Okay? So that's what you do here. Once you do that, once you declare this to be true, then you can sum over all the internal states, and that will give rise to a change in the mass as a function of these internal currents. Okay? J, this quantity is the loop current, okay, for the fusion, and this is the loop current. It's a bad notation, I'm sorry, but this is a loop current for the fusion, and this is the loop current for the fission. Okay? And only if that is non-zero do you change the mass. In equilibrium, okay, so here is the form of the loop current. Okay, the loop current involves basically it's the Kolmogorov loop condition. It's the rates that you go from on the right clockwise minus the rates that you go in the anti-clockwise. Okay, so if Kolmogorov loop condition is zero, is respected, then it is zero, and the current going through the loop is zero. So there's no change in the mass. Okay, so in the equilibrium limit, you don't have any changes in the mass. They can only come from active processes. Okay, so that's what I'm saying here. And what you do then, once you put in these ingredients, you can then look for how this kernel, both the fusion kernel and the fission kernel change as a function of m. And these are the forms that you get. So this is as you increase vesicle production rate, okay, you find that this is the form of the typical form of the kernels as a function of m. Fusion and fission, they don't intersect, so you won't get any structure formation. As you increase the production rate here, you find that you get curves which look like so. Okay, so these intersection points mean that there's a possibility of having structure formation. Yeah? Okay, so this is from the microscopic theory. Remember. Now that theory, of course, is very complicated. You'll see that there are many, many different parameters. So there's an effective simplified theory that you can write down, which looks like this. I mean, after all, this kind of curve can be mimicked by, by what? Any saturating kinetics, right? X divided by A plus X, right? So, in fact, you can make it more by putting in a possible Hill coefficient A. Okay. So now this is the equation that is derived from here for the average mass. Okay. We're going to now look at one cisterna only, one cisterna, getting material from there. And this is the equation that is derived with this is the time dependence of the average mass, averaged over the probability distribution that I talked about. And this is the fusion part, and this is the fission part. Okay? And this form then ensures that this is the form. Okay? The fusion, for instance, is this red curve, and the fission is the above curve. So you can see that for masses greater than this intersection point, fission is more than fusion, okay? And therefore this point is going to be stable. For masses below this, fusion is greater than fission, and so again that makes it stable. Okay? So this point here is a stable fixed point. Okay? It's a stable fixed point generated by this sort of feedback control of the fission and fusion process on the mass. Okay? Indeed, by changing parameters, you can allow for different kinds of topologies of intersection of these curves. And in fact, in one case, you can get two stable fixed points, like here. Okay? One for a very tiny mass, very small, and the other for a larger one. Yeah? And for those of you who know something about Golgi, this would be like the ergic. Yeah. Okay. So now what one does is takes these equations and asks what is the phase diagram in NV, namely the number of available vesicles at time t, NE, the number of available fission and fusion proteins at time t, and the mechanochemical feedback parameter. Because these rates, so that's what I forgot to tell you, that these rates here that I looked at are actually dependent on the mechanics or the tension of the membrane. Okay, so that's the feedback that one is talking about. So for instance, this rate where it goes from here to there, okay, depends on the feedback on the membrane tension. Okay, similarly here, to go from a flat membrane to a curved membrane, it depends on membrane tension. You can imagine that if the membrane is very taut, highly tense, right, then it's very difficult to form a bud, right? So you need the membrane to be floppy in order to form it. Okay, so that's where the elasticity of the membrane couples to the influences the rates. And it's that is responsible for the feedback control that I'm talking about. Okay. So that's as far as one single system is concerned. We've achieved the fact that it can form structures which are stable. Now you can ask what happens when you have two cisterna, and these are the simplified low-dimensional models for two cisterna, akin to what we had here. Okay, and in this case, what you have is that so this here is the one cisterna, this here is the second cisterna. Material comes from here to there. Okay, material can go back. Material can go from the first cisterna to the second. Okay, and material can go back, and then material can go from the second to the plasma membrane. Okay, so that's what describes these set of equations for M1 and M2. You can see that there are many parameters. It's more nonlinear, and for some of you, it may not be surprising that what you get here is an additional feature. What you get is a limit cycle in the cisternal masses M1 and M2. And indeed, there are two kinds of limit cycles you get. One is an in-phase oscillation, where M1 and M2 here are increasing and decreasing in phase. This corresponds to a periodic mass breakup, as seen in mitosis, right? As I told you, remember, in mitosis, organelles break up into tiny fragments. Okay, and for instance, typically in human cells, mitosis happens every 24 hours. Right? It's entrained by the cell, by the circadian clock. Yeah, and in another set of parameters, you find out of phase oscillations. That is, first you have a structure formation of M1, let's say. There's no M2, M2 is almost zero. Okay, then M2 forms and M1 goes to zero, and so on. Right? So this you can see that if it's coupled to space, will give rise to a traveling wave. And this is like a cisternal maturation model. Right? If you close your eyes and imagine, then this is what it is. Yeah. So, okay. So now this I think is a very interesting thing, because it's saying that cisternal maturation model and vesicle transport model are not two distinct models. The underlying physics involved in both of them are the same. They are just two different phases. Okay, so that's an important, I think, observation that comes here. Now, yeah. Okay, so now I'm going to end my talk and talk to you about ongoing work. This work that I talked about was carried out. The whole thing was carried out by my student Amit Kumar, and what he's doing now is to provide a detailed non-equilibrium phase diagram, which I was hoping to show you today. The possibility that if you have a time-dependent influx coming from the ER, because if you look at the influx coming from cells, it's not time independent. It also has a circadian rhythm. Okay, and so if you include that into these two equations, you'll get a third autonomous equation, and that of course gives you the possibility of chaos, right? And what is the biological realization of chaos is an interesting thing. It also gives rise to interesting features like Arnold's tongue and so on. And we're trying to understand, does this have any relevance at all to biology, or is it just a theorist's fancy? But that's what we are doing now. But in principle, it gives the possibility of entrainment with the cell cycle. Okay. We are also now doing stochastic versions of these to look at robustness to internal noise. And we are also studying control mechanisms. If there are sudden bursts of protein secretion from the ER, how does the system of Golgi, respond to that, right? Is there some way in which it can accommodate that and still maintain its identity? Yeah, the cell clearly does it. The question is, how does it do it? Okay. So now to end, I'll just the acknowledgements. The first work on extracting the kinetic parameters was done by my friend Pierre Sans and Serge Dmitriev, who was a student of Pierre Sans. The second work on the kinetic model to address structure formation and spatial control of Golgi was done by Himani Sachdeva, a student of Mustansir Burma. So we had a very long and nice collaboration for a few years. The last work that I talked about was primarily done by Amit Kumar, who is a student of mine, and by Suyupto Muhari, who was an ex-student and currently a faculty member in Pune. Okay? And funding for all this work is, as usual, through the Simons Foundation and the JC course. Yeah. Thank you. So, yeah.
Yeah, thank you very much for this beautiful presentation. I think there were a lot of things in there that are inspiring thoughts, things that you can all think about over lunch or on your way home in a few days.
Or while having a beer.
While having a beer. I'm not sure if we have beer here. That's another question. Anyway, we are a bit behind schedule, and I think most of you are hungry, but I don't think we should have no questions, because if there are questions, and I see one already, then we should have them now, okay? So let's have a few questions and then lunch. Please.
The Himanis. Yeah.
So there you wrote down the moment equation, right?
Yeah, exactly.
Like, did you look at the
Fluctuations? Yeah. We looked at the fluctuation.
Like, what is the nature of
Absolutely. Yeah. So, right, absolutely. So the paper has all that, but we also did a lot of physics papers on this, because that was the primary interest earlier, to look at the nature of the fluctuations. It turns out that when you have this systemmal progression model, that is, you allow for drift, then the fluctuations are system spanning. So they are very large fluctuations. And then they show intermittency and so on. So yeah, the nature of the fluctuations are very interesting. At one time we thought that maybe going along that route would be a nice way to predict whether you have systemmal majoration or vesicle transport, but I'm not too sure now whether that's a possible experiment that can be done. But yeah.
Okay. Other questions?
Yes. Reason? Why is there a pancake? So can you hold that question for tomorrow? Because tomorrow I'll show you that it's a pancake. I'll give you a physical mechanics model to show you that it's a pancake. Whether there's any reason, I don't know. Let me say now that it's a natural consequence of having fission and fusion. That the moment you take a membrane and allow for fission and fusion, it will spontaneously generate a pancake-like structure. Yeah. So no. So okay, yeah. Ask a question. Yeah. Okay, so let me just ask the first question. It's not, okay, there's been no study on how does the Golgi size and number increase with cell size. There's been a lot of study about how the nuclear size changes with cell size, and a lot of the things that Neer would have talked about, namely how does the centriole change, how does spindle change, how does so all these actin or microtubule associated structures, how they change with cell size. That's been well studied, especially recently, but there's no study of how does there is one study of how mitochondria changes, but there's no study about how Golgi changes, at least to my knowledge. Yeah.
Okay. One more.
You can ask now.
Now.
Yeah. Yeah. So if you stop system drift, so in the vesicle transport scenario, There, the steady state chemical composition doesn't vary with time. It's fixed. But when you allow for cisternal drift, then it has a much stronger fluctuations. Yeah, that's right. It shows the same pattern. All, yeah, exactly. Yeah. But then that will change with time. Yeah, 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 yeah. So that's exactly what was done in the last model that I talked about. So there, the fusion and fission rates, some of those microscopic rates depend on the membrane tension, and one has to write down a theory for the membrane tension. The simplest theory for the membrane tension is that it's related to the excess area. That is, if I take a sphere, right, where the area is related to the volume, then of course I can't make any vesicle, right? So I need to have a slight excess of the membrane area compared to what it was in the sphere, right? And then it can, so it depends on so in that way it depends on the membrane area. That's exactly the calculation.
Okay. I don't see very urgent questions anymore, and I think you're all hungry, so that's a good point to have our lunch break. So thank you very much again
Thanks.
for this presentation, and see you all at 2.